Wow, very cool to follow someone that does research on Antarctica. This is going to be also different than what we've been hearing about throughout this conference. I work in health informatics, and so I do not have the bacterial microbiome background, but I am trying to get into it more. I'm coming from the human genomics perspective, so I hope you can learn some cool things about that. So in addition to genetics and the environment, we know now that the gut microbiome contributes to human health. That's very known. So changes in microbiome composition contribute to dysbiosis and leading to lots of different health conditions in people. And so we're trying to look at the connections between the host genotype and the host phenotype and seeing that connection between our genetics and how they contribute to our health, and also our genetics and how they contribute to the composition of the gut microbiome, and seeing how these can all be related to each other. So we know that host genetics play a role in bacterial composition. There have been a number of genome-wide association studies that have been done that have looked at this. I'm showing an example from the MyBiogen genome-wide association study, which is the largest one to date, that has looked at, so you can see on the x-axis, we see chromosomes from 1 to 22, and on the y-axis, in order of from bottom to top increasing significance. And you can see that the gut microbiome abundance and presence are statistically associated with different bacterial taxa and genetics. So this is very interesting, but also it's really important to note that host population, so the genetic diversity of the actual people play a role in this. So if you look at some examples, you can see that people of Asian or Pacific Islander background have increased abundance of certain bacteria, while people of white or European ancestry backgrounds have increased composition of other bacteria. And finally, it's also interesting to see that you can stratify different conditions based on the presence or absence, not only of the bacteria in the gut, but based on their genetic ancestry or even self-reported race in this example. So as I mentioned, there's been a number of genome-wide association studies done. There were 11 that were originally done before 2022, sorry, microphone. And they are looking at the association again of the host genetics and the gut microbiome, and if there are any associations. And those that are significantly associated are being coined as MAVs, microbiome-associated variants. So a study back in 2022 looked at these MAVs and did what's called a phenome-wide association study and looked at all the diseases that are associated with these genetic variants or MAVs. And they identified 31 significant MAVs. And however, these were only identified in people of European ancestry. And if you can see from this graph, the majority of genome-wide association studies for the gut microbiome were done in populations from European ancestry. However, more recently, there were 2 studies that were done that looked at geographically, in the same geographical location, people from different genetic backgrounds. And so we looked at those additional MAVs that came up from those GWASs and included them to do an even bigger phenome-wide association study that I'll show you in a moment. So bigger is more important for us because when we look at genome-wide association studies or phenome-wide association studies, we need a lot of data to see these differences in these ancestry-specific backgrounds. So the original study in 2022 had a lot of people from 2 different ancestry populations, people from African ancestry in green and European ancestry in blue, and yet they were only able to determine a few MAVs associations with phenotypes in the European ancestry population. So we decided to look at many different biobanks from the United States. We have the Penn Medicine Biobank where I work, the All of Us Biobank, which is the National Institute of Health Biobank, and the Million Veterans Project. And so we were able to increase diversity in these microbiome PHOASs. And I know diversity means something different from all the talks that we've heard before, but here it's for people in addition to the diversity of bacteria. And so you can see we increased from 84 to 100% more people in this study, which is important for us. So first, I'm going to kind of go through the method very quickly. We took those 1,003 unique MAVs. We did phenome-wide association studies in both ancestry stratified and overall across the 8 studies from all these different biobanks. After running phenome-wide association studies or extracting the genotype by phenotype analyses from the 2 All of Us and MVP biobanks. And so we were able to get 877,000 people in this study. I'm going to first talk about the results from the overall meta-analysis. So we were able, by increasing the sample size and increasing the diversity of the population, we were able to uncover 32 MAV by phenotype significant associations. And this was 90% more than what had been previously reported. You can see on this quasi-Manhattan plot, which is the PHIWAS representation, where on the x-axis are 13 different disease groups from circulatory system on the left in red, which has many very significantly associated phenotypes to MAVs, and then all the way down to other symptoms on the right. And so if you look at this phenogram on the right here, you can see that where these MAVs are located in the genome. So you can see there's many cluster on chromosome 2, chromosome 2 and 6, and also chromosome 9 down here, and some others spread out across the human genome. And when we look at pathway enrichment analysis, many of these are linked to genes in, and are enriched in the immune system. So when I see where are these MAVs located in the human genome, I also am then able to take those genes and see what pathways they're associated with, and they seem to be enriched in the immune system, which tracks with the literature, and as well as some other pathways. So this will be explored further downstream. Now, when we look at the ancestry stratified results, we reveal significant associations, not only from the European ancestry results, which are significantly increased from before, but also in the African ancestry population, which is both, which is reassuring that there were some associations there to begin with, that the gut microbiome is not only important for people of European ancestry, but that there are actual differences between the population in addition to similarities. And so in the phenogram result here, you can see that the different shapes are representative of the different analyses. And so African ancestry is in circles, diamonds, or European ancestry and overall meta-analysis in triangles. There are only 3 associations that are unique to people of African ancestry. So I'm going to talk about those 3. These are actually suggesting novel connections that had never been reported before. So there's a MAV in the ABO gene that seems to be increasing bacterial taxa as well as decreasing levels of anemia in this population. This MAV over here is in a gene that has not really been fully understood, but we're going to follow it, to see what we learn more about. And then this MAV has actually been reported on a lot in the region of HLA, which is very important. And it seems to be involved with macular degeneration, which is very interesting. All of these had never been reported before. And, but this is interesting because these are all diseases that are, well, at least 2 of them are very prevalent in people of African ancestry. For example, anemia has a 3 times higher prevalence in people who are African American in the United States than white or white compatriots. And then glaucoma presents at 2 times higher rate in African American population compared to white or Hispanic population. So this is all very compelling for us to proceed further with. And then for the European ancestry specific associations, there was a few MAVs that converged on atrial fibrillation, which there's a lot going on with the connections that we're going to explore further down the line. So in conclusion, we showed over 300 MAVs linked to disease phenotypes, increasing evidence about these connections between the host genetics and the gut microbiome and how they contribute to disease. And we look forward to exploring all of these connections further and understanding how it all works. And if you have any bacterial insights, please let me know. So thank you to collaborators, our lab, and our funding, and all of the 877,000 participants who contribute their samples to research.
Thank you. Thank you. So we are a little bit behind on time, but I will allow one quick question, Johannes. Microphone, and then we can already set up the next speaker.
Okay. Oh, yes.
Yeah, thanks. That was a really nice talk. And I was wondering whether it's possible from your data or other people's data to distinguish whether the effects of these variants, these MVOs or whatever they're called, whether they are really through the microbiome or whether they have a direct effect, because immune genes obviously have a strong effect on all kinds of diseases.
Yeah, so we're going to be looking at that further. We'd like to be able to kind of like what you guys are doing is get, we would love to have individual level data to look at these effects. Right now we don't have that individual level data. That would be very helpful for us. But yeah, we want to know how to do that. That would be great. That'll be explored further. If you have suggestions, again, let me know.
All right, thank you. Right,